responsible for human disease. The members of Enterobacteriaceae, the member of Enterobacteriaceae is largest, most heterogeneous collection of medically important gram-negative roots or bacillus or bacilli. Currently, more than 50 genera and hundreds of subspecies and species have been identified. Enterobacteriaceae are a ubiquitous organism which found in soil, water, and vegetation. Members of Enterobacteriaceae are a part of normal intestinal flora of most animals and humans. They are classified based biochemical properties, antigenic structure, molecular analysis of their genomes, and also protein composition by mass spectrometry. The common features, they are gram-negative roots, they are facultative anaerobe bacteria, they ferment glucose and other sugar, they can grow rapidly on non-selective and selective media such as blood agar, nutrient agar, and McConkie. They have simple, on contrast, streptococci, in contrast of enterococci. Enterobacteriaceae have a simple nutritional requirement. They reduce nitrate to nitrite, they produce catalase, they are oxidase negative. Oxidase is a simple test for differentiating the members of Enterobacteriaceae from other fermentive and non-fermentive gram-negative roots, such as Pseudomonas and Vibrio. Vibrio and Pseudomonas are oxidase positive, but the members of Enterobacteriaceae are oxidase negative. Unlike other member of family Enterobacteriaceae, Pelizomonas is an oxidase positive. Pelizomonas belongs to the Enterobacteriaceae family. Pelizomonas is only genus that is oxidase positive. Antigenic structure of Enterobacteriaceae cell wall. The heat-stable lipopolysaccharide, or LPS, is the major cell wall antigen. It consists of three components: O polysaccharide, a core polysaccharide, and lipid A. Among them, lipid A has an endotoxin activity. It is responsible for many clinical manifestations such as thrombocytopenia, shock, fever, death, leukocytosis, and release of cytokine. The second structure is an H antigen or flagellar antigen. This antigen provides motility for Enterobacteriaceae. H antigen or flagellar antigen is responsible for motility. It is a heat labile. Some pathogenic species are associated with a specific O and H serotype. For example, E. coli L1-P5H7 is responsible for hemorrhagic colitis. Hemorrhagic colitis is an abdominal cramp. Hemorrhagic colitis is characterized by bloody diarrhea and abdominal cramps. The second antigen is a K antigen or capsular antigen. This antigen found only in certain species. For example, K antigen was observed in E. coli, and the BI antigen was observed in Salmonella enterica subspecies Enterica serotype type A. As you know, capsule interferes with the phagocytosis. The common virus factor associated with Enterobacteriaceae, endotoxin. The activity of the endotoxin is related to the lipid A component. LPS or endotoxin released after the lysis of the gram-negative bacteria, such as the members of Enterobacteriaceae. And many systematic manifestations such as fever, shock, thrombocytopenia, leukocytosis are initiated by endotoxin. Capsule. Capsule is another virulence factor. This virulence factor observed in some species such as E. coli and also Salmonella enterica. Capsule blocks the phagocytosis process. Antigenic phase variation. Expression, some antigen such as H antigen, O antigen, and K antigen is under the genetic control of organism. These features protect the bacteria from the antibody-mediated cell death. Type 2 secretion system. Some bacteria, such as Salmonella, Shigella, Yersinia, Chlamydia, have a common effector system for delivering virulence factor into the targeted eukaryotic cell, such as human cell. This system facilitates, accelerates transfer of virulence factor into the host cell. Sequestration of growth factor. As you know, iron is an essential growth factor for bacteria, but it is found in heme protein such as hemoglobin, myoglobin, or iron chelating proteins such as transferrin and lactoferrin. Bacteria produces siderophore. Siderophores are a small molecule. Siderophores are a small molecules that provide iron for bacteria. Nearly all strains of bacteria produces siderophore. Siderophore compete with the human and provide the iron for bacteria. Resistant to serum killing. Capsule protect the organism from serum killing. Another common virulence factor is antibiotic resistance. Antibiotic resistance is common in some species of Enterobacteriaceae, such as Klebsiella pneumoniae. Currently, Klebsiella pneumoniae is resistant to many antibiotics and create many challenges for clinicians. As per we used to mention, 50 genus and 100 species and subspecies have been identified. Most infections by members of Enterobacteriaceae are caused by few species. Some species are always associated with human disease, infections, such as Salmonella, Serotype typhi, Shigella species, Solei, Felixaceae, dysentery, and Boydii, and Yersinia pestis. These organisms are always related to the human infections. The presence of these organisms in clinical specimens is always considered significant. Other members such as E. coli, Klebsiella pneumonia, Serratia, Porotus mirabilis, Enterobacter cloacae, Morganella, and Citrobacter are members of normal commensal intestinal flora, but it can cause opportunistic infections. The first species that I will talk about them is Escherichia coli. Escherichia coli is associated with many diseases, such as gastroenteritis infection, extraintestinal infection, such as UTI, meningitis, and sepsis. Large number of E. coli are found in the gastrointestinal tract, but it can be opportunistic pathogen when the intestines are perforated and the bacteria enter the peritoneal cavity. Most strains of E. coli cause GI and extraintestinal disease. These organisms, strains of Escherichia coli have many virus factors. They are acquired virus factor from plasmid or bacteriophage DNA. Escherichia coli is the most common gram-negative bacillus or roots which isolated from patient with sepsis. It is responsible for 80% of all community-acquired UTI and many hospital-acquired infection. Most infection by E. coli, with the exception of gastroenteritis and neonatal meningitis, are endogenous. Because E. coli is a common commensal intestinal. That is, E. coli that are part of patient normal microbial flora are able to establish infection when the immune system are weak. For example, in HIV patient, in patient with war, in patient with trauma or immune suppression. Or E. coli can produce disease. For this reason, it is called an opportunistic pathogen. Can produce disease in patient with weak immune.
Doctor,
Five different.
What does sepsis mean?
When the organism introduced into the bloodstream, it is called sepsis. Five different groups of E. coli are responsible for gastroenteritis. Enterotoxigenic or ETC, enteropathogenic, enteroaggregative, Shiga toxinic, and enteroinvasive E. coli. EIEC, the abbreviation of EIEC, STEC, EAEC, EPEC, and ETEC. ETEC, EPEC, and EAC. The site of action of these species are small intestine, but STEC and EIEC targeted large intestine. Infections of small intestine by these species is characterized by watery diarrhea, but infections of large intestine by these strains is characterized by bloody diarrhea. Yes, this is bloody diarrhea. It is a watery diarrhea, but it's a bloody diarrhea. ETEC, the first species, ETEC. Enterotoxicity E. coli.
Is its infection just in GI system?
Yes.
Just there's
No. The common infection is the GI and also produces UTI, meningitis,
And sepsis.
sepsis.
Do you mean
This
that
species,
respiratory
this
system also?
species, this organism is found based on the presence of some virus factor are divided.
No, I mean the system that going to causes inflammation in it or infection. Just the GI system or the reproductive system and respiratory system also going to affect them.
These organs can produce many diseases. The common disease is a gastroenteritis and also produces meningitis, UTI. ETEC is one of the most common cause of bacterial diarrhea in developing countries. Infections are primarily acquired by the consumption of contaminated water or food, and person to person does the person to person spread does not occur.
It's not congested. Not congested.
Again,
Not congested.
congested.
Yeah. Doesn't spread.
No. This organ is primarily acquired by consumption or ingestion of contaminated food or water.
So it's
Yes.
inflammation, not infection. It's inflammation, not infection.
Yes. It is not person to person spread. And secondary diarrhea generally develops after 1 to 2 day incubation period and persists for average of 3 to 5 days. The disease by ETEC is characterized by these features.
Watery,
Non-blood diarrhea, watery abdominal gland,
cramps,
and
cramps
yes.
mean pain or
Yes.
yeah.
Migre belli.
Better.
Contraction of abdominal muscle.
Yes.
Sudden.
ETEC. Disease requires to the attachment of ETEC to the small epithelial cells. This bacteria attach to the small epithelial cells by colonizing factor. After attachment, produces two enterotoxin, heat stable and heat deprived enterotoxin. The gene encoding the colonizing factor and enterotoxin are encoded by plasmid. Colonization factors are subdivided several families. Among them, these families are the most common. Yes, colonizing factor 1, 2, yes, CFA means colonization factor attachment. After attachment by colonizing factor, ETEC produces two classes of enterotoxin, STA and STB.
Heat stable toxins
Yes, and
labeled
LT1 and LT2 are a heat-lapping toxin.
What's the difference between them? Oxygen? The difference between heat stable and heat lagging.
This is resistant to heat. This is
Not
susceptible.
resistant.
Yes, this is susceptible or sensitive to heat. STA, but not STB, is related to the human disease and is observed in 75% to 80% of.
Does that mean that STB is not cause any disease for human?
STA increase in CGMP, cyclic guanosine monophosphate, and subsequent hypersecretion of fluid, well as inhibition of fluid absorption. LT1 is more commonly associated with human disease. LT1 increase in cyclic AMP levels, resulting in increasing secretion of chloride and decrease absorption of sodium and chloride. These changes are manifested in watery diarrhea. Exposure to toxin, exposure to LTI also stimulate the release of prostaglandin and also production of pro-inflammatory cytotoxin, resulting in a further fluid loss. EPEC. Two groups of E. coli, which responsible enteric disease are EPEC and STEC. Both EPEC and STEC have many viral streams. The viral streams are located on chromosomal pathogenicity island. It's called locus of enterocyte efficient, or LEE. EPEC, based on presence or absence of EAF plasmid, adhesin factor plasmid, divided two groups, typical and atypical. Typical, EPEC typical, have this plasmid, but atypical do not have EAF. Disease is transmitted by fecal oral exposure to contaminated surface or food product. EPEC. Humans are only source for typical strains, EAF positive. But both humans and a variety of animals are reservoir of atypical strains. EPEC binds to the small intestine by adhesin. It called bundle forming peeling or intermittent. This is a virus factor. This organism, EPEC, binds to the human intestine, to a small intestine by adhesin. It's called bundle forming peeling or intermittent. After attachment, disrupt the surface. This is occur primarily in children younger than 2 years, and it is characterized by watery diarrhea because EPEC affected a small intestine, characterized by diarrhea, fever, and vomiting. EAEC.
Does it increase circulation of fluids? Increasing the circulation of fluid or exiting of fluid?
Yes. EAEC, enteroaggregative E. coli, is characterized by autoagglutination in a stack brick arrangement over the epithelium cell of the small intestine. It is an important cause of childhood diarrhea in developing countries, and it is associated with chronic diarrhea and gross filtration in children. This is why EAEC is characterized by these features: abdominal pain, vomiting, fever, inflammatory cells. This organ, this bacteria, binds attached to the small intestine by AAF, by aggregated adhesive fibrillary. EPEC binds by the bundle forming peeling and intermittent, but EAEC attached by AAF. Based on presence of different virulence factor, EAEC, we can identify EAEC, EPEC, STEC. STEC. All members of STA are defined by the presence of Shigatoxin 1 and Shigatoxin 2. The most common serotype related to human disease is L1-P5 Ingestion or conception of fewer than 100 bacteria can produce disease. Person-to-person spread occurs. Disease caused by STA ranges from mild uncomplicated diarrhea to hemorrhagic colitis with bloody diarrhea. STA initially attached by bundle-forming media and interin, and then produce this toxin. This toxin affects the colonic epithelium. Hemolytic uremic syndrome is the most serious life-threatening complication following STA infection. This disease, a disorder characterized by acute renal failure, thrombocytopenia, microangiopathy, and hemolytic anemia. HUS is a complication in 5% to 10% of infected children younger than 10 years. EIEC. EIEC strains are rare in both developed and developing countries. Pathogenic strains are primarily related to a few restricted O serotypes, such as this serotype. EAEC strains are able to invade and destroy the colonic epithelium and producing a disease characterized by watery diarrhea. A series of genes and plasmid mediate bacterial invasion into the colonic epithelium, like as STA, EIEC can produce hemolytic uremic syndrome. This is a complication of EIEC.
Are the stains needed to be memorized? The stains. The strains, do we need to memorize them?
Yes.
So everything you know.
Yes. Extraintestinal infections. E. coli is responsible for intestinal infection and extraintestinal infection. Extraintestinal infections include UTI, urinary tract E. coli. Escherichia coli is a common cause of UTI. Approximately 70 or 80% of UTI is caused by Escherichia coli. It is common cause of UTI.
But it's also can occur by another bacteria. Also may occur by another bacteria.
Another bacteria.
Yeah. Also may cause UTI.
Staphylococcus saprophyticus, Klebsiella pneumonia, and
Staphylococcus epidermis.
yes, Staphylococcus saprophyticus are a common cause of. Among them, Escherichia coli is more common than
the others.
yes.
Yeah.
Most gram-negative roots that produce UTI originate from the colon. They have several adhesions, such as PPD, AF1, 3, or DR. These adhesions bind to the cell of the bladder and upper urinary tract. Escherichia coli also produce hemolysin. Hemolysin lyses erythrocyte and other cells. Neonatal meningitis. GBS or group B streptococci and Escherichia coli are the majority of CNS infection in infant young. It is approximately 75% of E. coli strains possess common capsular antigen.
It's not
Septicemia.
common. It's not common to have an infection in CNS. It's not commonly to occur an infection in CNS. I mean, it's central nervous system.
Okay.
It's not common to find infection.
It was common.
Yeah.
Studies show that both
Yeah.
E. coli and GBS are responsible for neonatal meningitis. Septicemia. Septicemia, which caused by E. coli, originated from infection in the urinary or GI tract. Can you explain, Mom, understand this slide?
Sure.
Oh, sit down.
Okay. The bacteria ET.
You can see different group of STEM, EAC, EPEC, and ETEC.
ETEC, colonization factor antigens, is colonization factor A1, 2, and 3.
Okay. The antigen factor of ETC that binds to the small intestine
It's
are
colonization
colonizing
factor 1 till 3.
Okay.
Exotoxins, hate-labeled toxins, L1, stable toxins as TA.
Okay. ETEC are able to produce these exotoxins.
Hate-labeled toxins, LT1, and hate-stable toxins, STA.
Okay.
EPEC,
EPEC.
EPEC. It's the adhesion factor is EFP.
Bundle forming pili.
Intamine.
EFP bundle forming pili.
Bundle forming pili.
Okay.
Intamine.
And also intermediate.
Yeah. Exotoxins.
EPEC strain cannot produce exotoxins.
No exotoxins.
Fine. Okay.
EAEC. The adhesion factor is aggregative adhesion membrane.
Okay.
AAF 1, 2, 3. Intra-aggregative heat stable toxins, plasmid encoded toxins. This is exotoxin. And the last one is STEC. Bundle forming pili, intamine, the adhesion factor, and also the oxytoxins, Shigatoxin 1 and 2.
Doctor, if I could ask a question. In the EAEC from the exotoxins, it said plasmid encoded toxin. Is there another toxin that is?
No, thank you. The gene encoding this toxin located on the plasmid.
Can it be encoded in another place rather than
No,
the
the
plasmid?
toxin, the genes which encoding the toxin is located on the plasmid. Salmonella. Another species is
It's
Salmonella.
another.
I can read it if you want. I can read it if you want.
Do you have a question?
No. I'm saying I can read it if you want.
You want to explain something?
If
You
you
want to explain it?
Different.